critical need for right now is, first of all, monitor and understand what's in there. But second, with rotaviruses, is to decrease the load. It is critical because you can let the pig immune system fight whatever little amounts are there, probably even good for to stimulate immune response. But the load is the problem. If it's just too much through the roof of that rotavirus, piglets will get sick, and that's what you don't want.
Hi, and welcome to our latest edition of Swine podcast. I'm Jerry Purvis, your host. Today we have our guest is Dr. Anastasia Vlasova, and she is associate professor at Ohio State University, where she has worked in epidemiology and studying a lot of our health challenges. So I'd like, if you could tell us just a little bit about yourself and how you got to this point in your career.
Sure. Thanks for having me, Jerry, and thanks for introduction. So I was born into a family of veterinarians and scientists, starting with my grandparents in Russia. So, for me, it was like a no-brainer. I loved animals. I loved science. I loved research. That's all I heard since I was a child. So I was really, really happy to continue the family tradition. However, I have two brothers that are not as excited about animal sciences, and they're doing something else. So then, unfortunately, we had lots of challenges in post-Soviet Russia with science. The science is not funded well, so I really wanted to, at least to try how it is to work in a real nice lab and do real research. So, and the opportunity came to visit the Ohio State University. I came as a postdoctoral researcher into Dr. Safe's lab, Linda Safe's. And it was the real great opportunity for me because she's an excellent mentor, and she taught me a lot. And she actually enhanced my enthusiasm and, desire to continue working in science and help to improve animal and possibly human health, because a lot of research we do is actually fits the one health concept very well and can benefit both. So yes, but my focus animal is still pig, swine. So in most of the viruses we study, it's either coronaviruses, rotaviruses of swine, and some emerging or re-emerging pathogens.
Very good. That's an interesting story and pathway. We're glad you're here. We definitely have no shortage of health issues, particularly here. And that's our biggest struggle, I think, as a producer, is you're doing everything right nutritionally and feed, but, health is really is going to be an indicator of how successful you're going to be at the end. So that's, I think, a good topic today and one we can talk about. Maybe for our viewers, just tell us a little something about, we were want to focus, I think, a little bit on rotavirus. I think that's a very ubiquitous issue here in the U.S. and most herds are have issues. And it's like a gnat, it's just, we can't seem to really get ahead of that. So maybe just tell us a little bit about, the rotavirus and, maybe the epidemiology of that, and we'll talk a little bit more about it, how we manage it.
Yeah, sure. You already summarized the main problem is that the rotaviruses are really everywhere. I simply don't know a single swine herd that wouldn't have it. By the adulthood, mature age, all swine encountered rotaviruses at least once, twice, or possibly many more times. So it's virtually impossible to find serum-negative herds. And that actually alone represents a challenge for vaccination and other control strategies because, for example, we do have commercial vaccines against rotavirus A. And historically, they were there. They were targeting the dominant genotypes, G4 and G5. But they do include live viruses, live modified or live attenuated viruses. In those viruses, to induce immunity in swine, they have to actually be able to infect and replicate without causing much pathology. But because all animals are seropositive, especially sows or gilts that you're targeting with vaccination, so there is some degree of interference. So, and we did with Dr. Safe, like throughout the years, lots of research on, vaccination against the porcine human rotaviruses. And what we observed, the golden standard to induce or prime the immunity was the use of a live attenuated rotavirus A. So, but when it comes to boosting immunity, actually, our observation was that non-replicating vaccines better because that's how you bypass that interference by maternal immunity or pre-existing immunity. So because there, whatever the dose of the antigen you administer, that's what they get. And it's live vaccines, they rely on replication and some amplification of the virus when it enters pigs. So that's number one. Second, I would like to mention, when you say rotavirus, people think rotavirus A and there is vaccine, and why bother at all? But there's actually more to that than rotavirus A. There are rotavirus B, rotavirus C, and now emerging rotavirus H, which was already confirmed in the US, Brazil, Japan, and some other countries. So another problem that only rotavirus A strains readily grow in cell culture, and that's how you can manufacture vaccines, conduct pathogenesis studies, and all of that. Rotavirus B, C, and I think rotavirus H do not have really robust cell culture systems, and that's why the studies on those viruses are kind of lagging behind. We know much less about them. We don't have vaccines for them other than the new security platform from Merck that relies on mRNA technology. So yeah, that's in a nutshell. Like they're pretty much everywhere. They do differ slightly. Rotavirus A becomes a bigger problem around weaning, post-weaning, when the maternal immunity wanes. Rotavirus C, in contrast, affects the very young suckling piglets. And we are trying to get some preliminary data on that, because this pattern, when the A virus is not eliminated by maternal immunity, or at least not inhibited enough by maternal immunity, it tells us that maybe it learned how to benefit from some milk components. Because there was a publication from India, it's relatively recent, when they isolated a rotavirus A in a neonatal unit, like when it was really causing severe gastrointestinal disease in babies, in breastfed babies, which is unusual for rotavirus A. They isolated that virus and studied it, and sure enough, they found out that it likes milk oligosaccharides. So we're suspecting that something like this may be going on for rotavirus C. So yeah, the only system we can use right now is porcine small intestinal enteroids. It's a good system. You can pretty much infect enteroids with whatever virus can infect swine, but the downside is the same. If you need to isolate the virus, rotavirus C, for example, it's very challenging because all other viruses will be, or pathogens will be replicated in those.
We have a time and labor-saving product for you. AgriSlats by Healthy Farms is your solution. No more lugging jugs around the barn every month. With AgriSlats, you simply drop the slat through the floor twice a year, and it works to break down solids, reduces crusting and forming. To learn more, visit myhealthyfarms.com.
So it sounds like there's a lot of issues even with using vaccines to control rotavirus. So, just thinking about this seems something that we've just maybe seen in the last 10 to 15 years really, face an issue. Maybe it's been there all along. Why do you think it's more prevalent today? Uh, and we personally, we've got a very clean pig, and so I wonder if is there something there with interaction with other pathogens or something that we're seeing. Why do you think it's more prevalent today?
Yeah, well, I do not know for sure, but there are multiple theories that I could discuss a little bit. So, number one, for example, it's like immune evasion by the current rotaviruses. So, how it was, there were some dominant types of rotavirus A, for example, that I mentioned, G4 and G5. In the past, and they were dominating in the US herds for decades. Then the vaccines that were made by Merck, they were targeting those genotypes. So I think they created enough herd immunity, and of course, it inhibited spread and replication of these specific viruses. However, what it prompted, because the viral population always wants to survive, so they change. So the rotaviruses have a very unique mechanism of their reassorting their genome segments, creating new variants to evade this immunity. So, and I think different genotypes started emerging. Like, for example, in our lab, we work with the historic OSU rotavirus A strain that has a G5P7 genotype constellation. But we also work with a newer field strain, and that genotype is G9P13, and that one was isolated about 10 years ago. So, although they are rotavirus A, the cross-protective immunity is not very strong, and antibodies developed against one does not protect against another, number one. Number two, their biological features are completely different. For example, they differ drastically in how they interact with cellular receptors. The OSU, the G5P7 strain, likes sialic acids. If you remove them from the target cell, the infection rates drop. G9P13 is totally different. That one really doesn't like sialic acids and actually replicates better when you remove them, significantly better. So that's in vitro. But I think similar mechanisms are possible in vivo, and I think why it emerged, besides, I think, herd immunity, it is possible that, I don't know, maybe swine diets changed over the years. I don't know how much different they were 20, 30 years ago, but what might happen if the diet was different back then and now they're changed. So it may change the microbiome of the cows, the gut bacteria and fungi and all other stuff living inside the pigs. So, and rotaviruses, not only they interact with host cells and receptors, but they interact with all that microbiota. They adapt to that microbiota. And I think they also learn to benefit from that microbiota. That's how they survive and proliferate. Again, there was a study in humans that, for example, certain genotypes of rotavirus A are associated with certain microbiome compositions. So, which means they create some sort of symbiosis or synergism. So, for example, I think one possibility that the G9P13 strain learned to benefit from some bacteria living in the gut that can remove sialic acids for it. Because rotavirus, unlike influenza, for example, doesn't have its own neuraminidase to do it for itself. So it has to benefit from something external. So that's another possibility. What else can it be?
So it looks like it sounds like it's a constantly changing. And you mentioned diet, maybe the microbiome. There's some cross talking there with bacteria, which brings up a point. Is there anything you think as far as the environment or managing that we're doing differently today? I know a lot of these producers are really pushing, wean age. They're pushing, throughput, disinfecting between, groups. Are there some things you think maybe we're missing the boat and not managing today? like we used to, possibly?
Well, that's another excellent point. Yes, I think the swine herd management practices changed drastically. I think the whole pork production industry intensified tremendously in the past few decades, like 2 decades. So I think maybe, yeah, one thing I noticed for sure, for example, now we're getting a lot of litters for research that, like, each sow seems to be bringing more piglets. We've been working with litters of 8 to 14 piglets for years, and then suddenly, like last 10 years, we're getting litters 14 and to 21. So I think that even the fertilization breeding approaches changed quite a bit. So they probably treat them with different hormones, and that alone can affect the microbiome. It can affect the pathogen susceptibility because all this transitional the transitional period in cells like periparturient, it increases susceptibility to different pathogens. So I imagine that other sex hormones can cause similar effect.
Yeah. So if we I know you mentioned litter size has gotten bigger. Do you think there's some correlation there in colostrum intake? Or I know you mentioned earlier how is that passive immunity a little? Maybe they get some impact if they get colostrum and start nursing pretty early, pretty soon after birth.
Yeah, that is a critical point. If we're talking about newborn neonatal piglets, they have to start suckling colostrum. The gut closure has to happen, and barrier function has to be established. They get this passive immunity. They have to be able to nurse. They have to be able to nurse efficiently. Like, so, for example, if there is a runt in the litter, it just simply doesn't get enough milk. So, yes, all of those things. Obviously, too early weaning would not benefit them in that respect. Also, substituting sow milk with formula, it's going to be another downside, and I don't know how much of that producers are doing now. Sometimes sows can be aggressive, and don't nurse well, and they have to substitute with the formula. So all of that, I think, matters.
Yeah. So so again, we're talking about things we can control. That if we get those pigs nursing. Typically, just going back clinically, what are the signs you can see? It seems like it's really across the board is how virulent these strains are. What do you typically see as far as mortality, morbidity when you have these outbreaks?
You mean for rotavirus A, right?
Rotavirus, yes.
Rotavirus. So generally, if it's an uncomplicated infection, means other pathogens are not involved, I think the mortality due to rotavirus is below 20%, 10 to 20%, but I don't think it goes above that. However, quite often rotavirus infections can be followed by secondary bacterial infections because rotaviruses have mechanisms of transiently suppressing host immunity. They have several non-structural proteins that work specifically to downregulate that, which helps with their own replication, but then the effect lasts a little bit longer and other pathogens can benefit from it. So you really have to account for that as well when you're, evaluating the impact of rotavirus infection.
So it's really holistically, what is that animal's, challenge? It could be multiple challenges, other pathogens that could influence what is the.
Right. And it can be, because conventional pigs, whatever is present in a herd, piglet is going to get it, right? So, and just however clean the herd is in general, not only rotavirus wise, but, we cannot keep it out.
Right.
But another thing, is that the dose is important because the immune system can counteract some small amount of any pathogen, right? But when it exceeds that threshold, of course, it becomes a problem and a clinical disease develops. So besides secondary infections following rotavirus infection, we can have concurrent infections with E. coli and all other stuff, and that that can become a big problem, and then the mortality rates can be very high. So it's important to keep your herd as clean as possible. And sometimes, you have to use antibiotics even if they don't do anything directly to rotavirus, but for bacterial pathogens that are associated with rotavirus infection, it's important. So we also in our own studies, we observed that rotavirus C at least, pathogenicity varies per strain, per genotype. So some of the genotypes are naturally more virulent than the others.
So, as a producer, what are some of the maybe best practices we can use to manage? Just as far as how we manage the farm. What are some key things that we can do to at least maybe minimize the impact?
That's a great question, $1 million question. So, I think many of these practices are already in place. Of course, cleaning in between, all in or out, disinfecting well, trying to keep out as much pathogen and prevent introducing new animals around parturition. Um, Feral facilities have to be probably the cleanest because that's when the rotavirus can cause the most impact. I think vaccination very close to parturition to boost immunity, lactogenic immunity specifically, is also a good strategy. We all know that feedback method is used, which gives you an advantage of targeting the pathogens that exactly present in a herd. But of course, there is always a downside of introducing other pathogens in your ferrying facilities, which is probably less than desirable. I know different companies are trying to also develop somewhat customized approach. Obviously, Merck with their security platform. I know some other startup companies that they actually conduct a mini epidemiological study to see exactly what pathogens are present in a herd at this moment. Then, like if it's rotavirus, what genotypes of that rotavirus, and then they generate at least VP4 and VP7 proteins of those rotaviruses in a baculovirus system or any other expression system. And then that way they can also target directly what's in the herd.
It sounds like diagnostics is pretty important as well.
It's critical. I mean, if you don't even know what you have out there, it's just like I would do NGS on every sample, but it's of course tremendously expensive. So what else? I think with rotavirus, what we are doing right now, we are very interested in rotavirus extraintestinal replication because there is a lot of emerging evidence that rotavirus, it's not a strictly enteric pathogen as most people think. Of course, the major diseases, gastroenteritis, diarrhea, and this and that. But what we learned from human clinical studies, from our own preliminary studies, from other people's studies, field studies in swine, that rotavirus goes beyond the gut. We know it targets other organs like liver, salivary glands, mammary gland, and the lungs specifically. Most of those organs have no pathology, no gross lesions, no histopathology, but it seems there is some degree of pathology present in all our notobiotic pig studies with different rotavirus genotypes. So they look like multifocal hemorrhages, and we've been looking at it for about 20 years. We just couldn't believe in it, but now we just decided to move on with those studies. And quite often, if we do see those lesions, we do detect rotavirus there. So another bulk of data is on how rotavirus is spread. Again, fecal-oral route is the major route, and everybody knows about it and targets. But what we think, it also spreads with aerosols. And that's why it's like near impossible to keep it out. We've seen how rotavirus was spreading from one research facility, research building here to another, where only our animal care personnel had access. And we were like, no way. They change speed, they disinfect their boots, they do everything, but rotavirus is there. So, and I think that needs to be looked into very carefully, because if our control measures are inadequate, if we're not considering another major route of transmission, we're not doing the best job possible. So, but first you need to confirm it experimentally. That's what we want to do in our lab, to see what the impact of aerosol transmission. Would the infection be as, efficient, and transmission as efficient as fecal oral, and so on and so forth. Because again, clinical studies in humans, in human babies, children, a bit ahead of the swine industry, and they already shown that they can isolate rotaviruses from aerosol samples in hospitals. Someone already did a research in mice aerosol infection, and yeah. So if it gets in the lungs, in the respiratory tract, there is no way for it not to spread via aerosol. But we probably would need to update the management strategies, at least, for ferrying facilities, I don't know, air filtration or more harsher disinfectants. I don't know, but maybe mask wear, not for pigs, I mean for humans.
Yeah, so it sounds very discouraging. It seems like maybe we're not even, understanding all the routes of transmission, and we're maybe not even covering, the aerosol portion there.
Discouraging, but I mean, at least, it's important to rule it out or confirm.
Right. Yeah.
If possible at all, let's look into it and put a check mark whether
Yeah.
it does or doesn't.
Yeah, because if we're not, maybe as you said, maybe filtration, maybe there's some other things that we could really, lock it in a little better and reduce the transmission and spread. But we definitely need to know. We need more information for sure.
Yeah, and we did know for years, I mean, decades, that rotavirus is shed both fecally and nasally. So, and from there, it's not too far from aerosol transmission.
Yeah, it's just like, kind of like we've realized now PRRS can be transmitted through feed. So now, we used to think about the feed mill side of producers was probably pretty safe as far as eliminating as a transmission vector. But now we got to rethink and maybe change our plans and take that into consideration as well. So these viruses are smart. They're going to change, and they're going to try to survive. So they're going to try to get around all of our paths. Now, you mentioned cleanliness. Do you have a particular disinfectant that works better? As far as I know, you're talking about the farrowing room is pretty crucial, having a very clean, environment there.
Right. Yeah. So I'm not sure what's available to the farmers, but we here in our research facilities, when we disinfect between the trials and experiments, we heavily rely on spore cleanse and Vercon. But what we realized that some of our protocols, when we had those contaminated facilities, so the time of contact is critical. So when you just go strictly by what the bottle says, like 15 minutes of contact time, and then you can rinse it off, sometimes it's insufficient. And we were expanding it to 30 and longer than that, because rotaviruses are very stable. They're non-enveloped viruses. They're very stable in the environment.
Yeah. So, and this is something I've been thinking about a while. It seems like we're really pushing throughput through our barns, and we really don't have the downtime. Is there some advantages, particularly to rotavirus? Let's just say you disinfect the barn, you dry it. If you let it sit for a couple of days, what are some, do you get some benefit there on some kill? Is there a magic number of how many days, you need that barn to sit empty that's disinfected?
Yeah, I don't think there's a trick or magic number right there. Like, apart from the extended contact time and maybe repeated treatments with, disinfectants, I don't think just sitting would help much, because, for example, coronaviruses, they're very sensitive to the sunlight or UV. Viruses do not seem to be that way. So maybe targeting broader acting disinfectants is a good idea, because if you want to treat for rotavirus, you may just get away with vercon or spore cleanse, but maybe harsher like bleaches and bleach-based to kill all the bacteria too, or as much as possible, at least even environmental bacteria, because it seems that rotaviruses can benefit from bacteria being present on surfaces because they can bind to some bacteria, and in some cases it stabilizes rotavirus virions, norovirus virions. So yeah, I don't think there's a magic trick, but because the problem is so comprehensive, I think the approach should be very comprehensive.
Yeah. So it sounds like it's if we can just reduce that pathogen load in general, whether it's bacteria and viruses, we're gonna see a better response and better impact. You
Yes.
know, on
Another
that.
approach we were investigating in one of the projects I had in my lab, we were trying to see if it's possible to create a broadly protective probiotic. Because generally probiotic approaches that are like everybody knows about lactobacillus and bifidobacteria and that they're beneficial, and then you do one trial in humans or pigs and see great protection, and someone tries to reproduce and it doesn't work. And because it varies with the probiotics type and strain. It varies with rotavirus genotype, or let alone different rotavirus species that we discussed earlier, like A, B, C, and H. So we were trying to evaluate the component, because everybody knows that probiotics improve gut integrity or gut barrier, that they cause some degree of immune modulation, boosting the immune system. But we were looking specifically at the mechanism of how certain commensal or probiotic bacteria can mechanically bind rotavirus particles and inactivate them that way, or how they can block rotavirus attachment sites on epithelial cells, because for rotaviruses, although we do not know the specific bona fide receptor, but we know that they do interact with sialic acids on cell surface, they do interact with histoblood group antigens on cell surface. So targeting all those glycan interactions, so some bacteria learn to express those histoblood group-like antigens on their surface, most likely to evade being recognized by the host, and so evade the host immunity. But because of that, they can bind rotaviruses. We were trying to assemble a cocktail of bacteria that express those different histoblood group like antigens, and they would be it's a composition of several bacteria, so in the specificity of these histoblood group antigens would be different. So, and we were trying to see if they would be able to consistently reduce the disease and infection by different rotaviruses. A and B and C were included in our study, and it seemed that there is some added effect if you compare to the bacteria that do not express those structures. So yeah, besides keeping your farm clean as possible, facilities, maybe more systematic approach, vaccinating when it's needed, supplementing probiotics, and the correct probiotics, not only the ones that improve feed conversion, or how do you call it, performance, reproductive performance, and this and that, but also the ones that have very good and comprehensive anti-infectious characteristics.
Right. So it sounds like maybe we're targeting the bacteria, and that's going to give us a boost there and some assistance there, possibly certain bacterial strains. So that microbiome, that seems to be an opportunity, maybe a challenge that we learn more and we're able to combat indirectly.
Yeah,
virus.
I think we would be able, if we would learn a bit more about interactions of rotaviruses with bacteria, their interactions with milk oligosaccharides and other prebiotics, or the components that bacteria like to feed off. So maybe we could create some pretty evidence-based probiotic or symbiotic or even postbiotics. If you don't want to introduce live bacteria, you can use postbiotics that still can benefit. So,
And going back to a point, I wanted to talk a little bit about. You mentioned feedback, and I know a lot of people use feedback, particularly PD. And you said that somewhat of how does that option work? Will we see some success with that, or what are some of the disadvantages per se?
yeah, I think feedback has the benefit of targeting the range of pathogens present in a given herd, which is great and which is your ultimate goal, right? Because homologous immunity is always better than heterologous, even though broader is good, but not always achievable. So, but the undesirable effect is that you can introduce additional pathogens, additional pathogen load at the time you don't need it, at around
Right.
50 and stuff like that. So I would say probably every farm needs to monitor what is there in their, like, fecal material from the pigs. And I would say if there is suspect of PRRS or PDV or some real epidemic viruses or the ones that can suppress immunity, maybe not the best in that
So
sense.
it gets back to diagnostics, just knowing what you have and what are the challenges, your pathogens you're facing is
Yeah,
important.
I think diagnostic and epidemiological surveillance are critical components. And I think also, like, I noticed that many veterinary diagnostic labs are trying to create some sort of networks, inter-university, interstate,
Yeah,
and exchange the information because, sometimes it helps to know what's in your farm. Sometimes it helps to know what in the other
yeah,
farm too. So,
yeah. Collaborating there, I'm sure will be beneficial as we communicate. Yeah. So in your mind, do you think this is something we can eliminate at some point, or is this something we're just going to have to live with?
I think, well, yeah, I'm kind of very realistic person and very practical. So at this point, I wouldn't even dream about eliminating it. I think the critical need for right now is, first of all, monitor, and understand what's in there. But second, with rotaviruses, is to decrease the load. It is critical because you can let the pig immune system fight whatever little amounts are there, probably even good for to stimulate immune response. But the load is the problem. If it's just too much through the roof of that rotavirus, yeah, piglets will get sick, and that's what you don't want. And then it will start the vicious cycle. They get rotavirus, and they get secondary bacterial infections or other pathogens, like even viral infections because of immune suppression. And yeah, so first I would try to achieve the goal number one is decreasing the load. And maybe limiting the transmission if there is a possibility of airborne spread.
Yeah. So it sounds like we're going to do the best to get your best immune response in that pig, whatever um, to boost that immune. But at the end of the day, it's that load, that viral load, and trying to minimize that so that pig naturally can fight that off uh, with immunity and reducing that load in the environment. So.
And maternal immunization, I think it's also a very viable approach. But again, it matters when you vaccinate your pigs. We did conduct several trials for PDV and rotaviruses in South, and seems that the last trimester is a good target because unfortunately immune boost does not last very long. But yet you need to allow a couple of weeks to, immune response to occur in sows to boost it in piglets, to pass it on to the piglets. So yeah, it has to be very evidence based for everything. It's some vaccinate, but do not vaccinate it at the right time, and then it doesn't help. And I think the very big attention should be paid to the guilt. because that definitely the weak, how do you go, block in the chain or whatever.
Yeah, very limited immune uh, system to pass on to the piglets for sure with gills. Yeah. So, well, I think you're giving us a lot of good. It sounds like there's a lot of things we can do to control. How impactful this is. Maybe we don't eliminate it, but we can minimize the impact if we control some of these the environment and making it very clean and reducing that load, as you say, that piglet is exposed to. So I think we've got. I think the viewers got some things we can go back to the barn today and just it's almost it's like it's the things that we know that we need to do, but sometimes we don't really focus and prioritize those things. So maybe it's just getting back to what we know works and making sure that those things are doing and hopefully, like you said, with the aerosol, we learn, we figure out maybe what are some other things we're gonna have to do to really tighten up our barriers for sure. So it's very good. Last before we leave, I was thinking about what are some of the emerging viral, maybe talk a minute, maybe some emerging viral pathogens that you see.
Yeah, well, there's always something that emerged. So if we get away from rotaviruses, of course, there's always something brewing in terms of coronaviruses. You probably heard about, the one in China, SADS, swine acute diarrhea syndrome. Coronavirus seems to be pretty recently jumped from the bats and causing a lot of trouble. So, of course, everybody knows about the threat of African swine fever virus, and I'm so not looking forward to that one.
Yeah, yeah.
Yeah, because probiotics is probably going to be useless for that one.
Yeah, I'm really. I think we obviously maybe have done a good job or been very lucky keeping it out here out of the U.S. But for sure, that's a really big black swan that hopefully we can. Hopefully, the things we're doing are working, and that we just be vigilant, and making sure we're taking care of every person is doing the best they can to keep that virus out. So, not looking forward to that. Yeah, hopefully that's something we can stay away from for sure.
I hope. I know several research groups are trying to work on vaccines and have them ready, stocked up in case it will arrive in the U.S. because with the amount of trade and moving the virus being so widespread there, because it was staying out of European countries for decades, but then once it moved fast and got
Yeah, it's like I said, we've got a big barrier of water, that keeps. But like you said, there's so much trade, so much travel, and all it takes is just one sample, and the virus is so. I mean, it's just so tough. It's
oh
just able to withstand and live in conditions and be dormant, and then so it's really a beast
yeah.
of a virus for sure.
Yeah, with that one, we may start looking into genetically resistant.
Yeah, yeah, that may be our best bet there for sure.
Yeah, I'm gonna mention that. Yeah, African swine fever virus can survive in a piece of sausage like for weeks. So
Isn't that amazing? Yeah. I know our farms, we don't allow the employees to bring in, you know, pork products just for that reason. Yeah, it's amazing what we learn about these viruses. And I guess that's just their ability to survive and evolve and mutate and be able to, develop all these bypasses to all the typical barriers we have. So it's a little bit scary for
but
sure.
better be prepared, scared and prepared than not, right?
That's right. Yeah, prepare it and hope nothing happens, but be prepared if it does happen. So
That's right. That's right.
I think we're doing a good job of that, and hopefully that'll continue. Well, we're nearing the end, and we've got the three questions we want to ask.
It's time for our famous three. Sunai is a professional solution provider for enzymes, probiotics, and functional additives. We offer customized formulas to target nutritional issues in livestock and maximize the utility of animal feed. Find us at sunhy.com, spelled SUNHY, for more information.
First is, what is your favorite swine resource?
Oh, well, yeah, I think there is the pig side. I think I like going to Swine Health Information Center. But just recently, I became aware of all the podcasts and Swine Disease Eradication Center and all of that. So that's really a good source too. Um, yeah, that's about what I know. National Park Board, we always go there. They follow up pretty closely on all, emergent diseases, and they always request the feedback. So I like to interact with them as well.
Yeah, it's amazing what the access to information we have today.
Oh,
Uh,
I just love it.
yeah, you can just Google something in a few seconds. You get an answer to your questions. So it's amazing.
Unless it's commercial secret. It's not that easy to find, for example, on the Merck website, what specific antigens are included into their vaccine. That doesn't pop up right away, but I understand that.
Yeah, we're spoiled. We get the answer just in a few seconds. We don't really have to do much work.
Yeah,
Uh, dig out the answers.
it's way easier nowadays than it was like 30, 40 years ago.
Exactly. Now, Dr. Vlasova, what do you, just thinking back, who were maybe some of your best or most important influences on your career?
Oh, that's definitely Linda Safe. Because not only she's a very, knowledgeable scientist, but she's very also courageous. She's an excellent role model. I mean, and she still has so much enthusiasm and passion for science. So yes, she would definitely be number one. There are others too, but yeah, I would put
Yeah.
her.
And that kind of rolls into my next question. And you mentioned passion, but what are some of the characteristics for people in our industry, in your field? What are some of the characteristics that make people, of those successful people, researchers?
Well, yes, I think you named one, passion, because you cannot do good science, good research, or taking care of animals if you're not passionate about it, because it's difficult. It's a lot of work. It's constant work. So passion definitely should be there. Then constant critical thinking, searching for information, new information, refreshing your knowledge, and really be critical about it. Don't like get stuck about like, well, I knew everything about rotaviruses 40 years ago. No, you have to keep studying them all the time and never assume that you know everything, or at least everything you needed to know. So studying, and critical thinking, updating your knowledge. I think constantly requesting feedback from like your critical points of information, veterinarians, farmers. Because I really feel strongly about One Health concept, because I don't think there are viruses that are purely human or animal. I think their zoonotic potential is way underestimated, including rotaviruses and coronaviruses, and maybe pestiviruses that never been
Hmm.
found in humans, but we're finding them now in those animal pestiviruses. So you really, sometimes I go to the clinical and human studies to inform what we need to do in swine industry or for animal health, because it's still different how humans are treated or babies are treated in piglets. So sometimes they have access to more resource, more knowledge, more clinical observations, like case studies. It's very important too. So you need to have access to that knowledge, and you have to be able to analyze and synthesize and come up with something.
Yeah, that's very good. I think good point, particularly on knowledge. We think we know as much as we need to know about something, but I find myself going back and maybe a technology I think I understand. I go back and look and I just understand it a little better. Maybe I pick out some point that I hadn't thought about, and then that changes my whole thinking about maybe how I attack some problem. So I think that's a good point. We all just constantly finding out new information, but maybe doubling down on the things that we know that we think we know, but maybe we need to know a little better. Helps. Yeah, very good. Well, uh, Dr. Vlasova, I really enjoyed our time here, and I think there's some very valuable advice here on rotavirus, and like I said, it's ubiquitous across all production systems, it seems. So I think this is something that the viewer can take today and go back to their farm and apply some of these, maybe rethink how they're doing things and manage this a lot better. But hopefully, keep up the good work. And because as you say, these viruses are just going to keep coming at us. We think we eliminate one pathogen, and there's always another pathogen in the throes ready to emerge sometimes. So I think there's not ever going to be a time when we eliminate all these things. We just got to learn how to manage them.
Thank you very much. I enjoyed very much talking to you and your thought-provoking questions, and of course, your insights from the practical side. It's very important for us as well.
I appreciate that. We appreciate you coming today, and look forward to more good work.
Sure. Thank you very much.
All right. Thank you.